Swayam Prabha. Digital India. Educated India.
Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the course Enzyme Science and Technology, we are discussing about the different aspects of the enzyme. And in the current module, we are discussing about the different types of assays to measure the enzyme activity. So in this context, if you remember that we have said that the enzyme is playing a crucial role in running the anabolic reaction and as well as the catabolic reaction, and both of these reactions are important for the survival of the host. And if you want to monitor the anabolic reaction or the catabolic reaction, you have to develop a suitable assay system so that you can be able to measure the activity of an enzyme. In this context, we have discussed so far what we have discussed about the photometric assays. And in the photometric assays, we have discussed about how you can be able to measure the absorbance of the system and how you can be able to use that information to calculate the concentration of the substrate or the concentration of the product utilizing the Beer's-Lambert law. And then we can also be able to use the fluorometric assays, where you can actually be able to use the fluorescent substrate or the fluorescent product. And that's how you can be able to use the photometric assays. In today's lecture, we are going to discuss about the chromatographic, we are going to discuss about the radiometric assays and as well as the assays, how you can be able to perform the assay of an enzyme in the using the gel electrophoresis. So let's start discussing about the radiometric assays. So as the name suggests, the radiometric assays mean the assays where you are actually going to use the radioactive or radioactivity as the monitoring substance, right? Just like as we discussed about the photometric assays, we were discussing about the absorption and excitation and all those kind of phenomena. In the radiometric assays, you are actually going to measure the radioactive counts. And remember that the radioactive assays are going to be done under the strict guidance or the strict rules and regulations, so that you should not have a radioactive wastage and as well as you should not have the radioactive spillage. And I have already discussed all these aspects, what are the different types of precautions you should take when you are going to do the radiometric assays in my another MOOC course called Experimental Biotechnology. So in that case, we have discussed about the different types of precautions and other kinds of the things what you should actually be able to wear. So for example, when you are doing radiometric assays, you always have to have the radiometric radioactive batches, right? And you should also use the lead walls and other kinds of shields, and so that you should actually be able to protect yourself while you are doing radioactive assays. So as the name suggests, radiometric assays means you are actually going to use the radioactivity. So the radiometric or the radioactive enzyme assays, which use a radioactivity labeled substrate, have been used successfully for many years to measure a wide variety of enzyme activities. The application of radiometric assays were initially restricted because of the limitation in the commercial availability of suitable labeled substrate and the scintillation counting instruments. Radioactive enzyme assay have successfully been used for enzyme substrate labeling with a substitute like tritium, 14C, 32P, 33P, 35S, and I-125. So these are the radio labeled nuclei because they are actually going to give you the radioactivity, and you can be able to make the substrate along with the either the tritium labeled hydrogen, tritium labeled 14C carbon, phosphorus, and so on. Okay, so depending on what kind of atoms are present in your substrate, you can be able to use either of these. The configuration of radioactive assay will in most cases be determined by a satisfactory quantity method of separating the labeled product from the unreacted substrate. With homogeneous radiometric techniques such as scintillation proximity, this is not a longer a issue actually. So in one of the major chunk or major task of performing radiometric assay is that you are actually going to have the separation of unreacted substrate from the labeled substrate or the product. But with the help of the scintillation proximity assay system, which we are actually going to discuss in this particular lecture, there is no need to purify actually. Radiometric assay has highly sensitive, specific, and absence of interference make them ideal for such high throughput screening applications. In the pharmaceutical industry, where there is a large and growing demand for the screening assay in the early stage of development. So, radiometric assays are very popular in the pharmaceutical industry, where you would like to use these kind of assays in the high throughput screening to screen the different types of inhibitors or drug-like molecules. When you are going to do the radiometric assays, you are actually going to use the different types of associated techniques. So radiometric assays are typically based on the conversion of radiolabeled substrate to the labeled product, the availability of suitable labeling substrate, and a simple and rapid method of quantitatively separating the product from the unreacted substrate are the two major requirements of a radiometric enzyme assays. So when you want to perform the radiometric assays, you are actually going to use all these assays or all these techniques. You are going to use the ion exchange techniques, you are going to use the precipitation, solvent extractions, paper and thin layer chromatography, electrophoretic method, and as well as the scintillation proximity assays to purify the labeled product from the unreacted substrate. Because unreacted substrate is also radioactive, your labeled product is also reactive, right? So that's why you are supposed to purify the labeled product. Scintillation proximity assay. The scintillation proximity assay is an assay where you are actually going to, where you have the beads, and these beads or the scintillation proximity beads, and these beads, when they are receiving the beta particles from the radioactive material, they are actually going to give you the light. Okay. In an ideal setting, when you want to do the radioactive measurements, what you are going to do is you are actually going to take the radioactive active nuclei and put it into the scintillation cocktail, and then you are actually going to put the vial into the beta counters or the gamma counters, depending on the type of radioactive nuclei what you have used. But in the case of scintillation proximity assay, you are actually going to have the SPA beads or scintillation proximity beads, and these scintillation proximity beads, when they are actually going to receive the beta particles from the radioactive nuclei, they are actually going to give you the light. And this excitation of scintillation proximity assay, scintillation proximity beads depends on the distance of these beta particles. So it's actually going to give you a very specific signal. What is the application of the scintillation proximity assays to enzyme assays? So SPA can be used to determine the catalytic action of the enzyme. The majority of the SPA enzyme assay uses the biotin-related substrate that can either be immobilized or subsequently captured by the streptavidin coated SPA beads, right? So you can have the SPA beads, which is actually going to be coupled with streptavidin, and you know that the streptavidin has a very strong affinity for biotin. So if you have the biotin on your substrate, as soon as the substrate is going to be captured by the SPA beads are actually going to start emitting the lights, and that's how you can be able to quantitate that light, and you can be able to calculate the concentration of the enzyme. You can actually be able to use the SPA based enzyme assays to measure the activity of hydrolases, transferases, polymerases, kinases, and the technique is applicable to the tritium leveled, iodine 125, sulfur 35, and the phosphate 33 labeled substrate, and optimization is required as with the other enzyme assays. The conversion of the substrate to the product is tackled by designing the assays to remove or add the radioactivity in relation to the component captured on the SPA beads. The process can either involve the removal of radioactivity by the enzyme, resulting in a decrease in the SPA signal, or the addition of radioisotope, resulting in the increase in the SPA signal. So you can actually be, just like as we discussed, right, either you can be able to measure the substrate concentration and you can actually be able to monitor the decrease in substrate concentration, or you can be able to see the increase in product, and then you can actually be able to see the, so that's why the SPA beads can be is a very convenient way of measuring the either the initial concentration of the substrate, and then you will going to see the decrease in initial concentration of the substrate, or you are going to see the increase in the concentration of the product. In all cases, the discrimination of the product from the substrate does not require the component to be separated, and SPA is a homogeneous technique. This has the advantage in some instance that incomplete recovery or detection of the product is not an issue. When compared to the methods such as precipitation and filtration, SPA enzyme assay exhibit high precision and reproducibility when a large number of samples must be analyzed in a short period of time. SPA is a very powerful technique. Then so this is what it is showing, is that how you can be able to use. So either you are going to have the increase in the signal, or you are going to have the decrease in signal. So the signal decrease assay format is appropriate for the hydrolytic enzyme that act on a single cleavage site on the substrate of the choice. The substrate is designed with a bead binding site and a radiolabeling site separated by an enzyme cleavage sequence. Most signal decrease assay can be designated as a solution phase or solid phase assay, but not all are solid phase compatible. For example, you can be able to use the SPA based protease assay to measure the Hi1 protease. Okay. Then we can also have the signal increase assay. So in a signal increase assay, you are going to first, this is good for the transferases, polymerases, and the capture format. Okay. So signal increase SPA format has been used biotinylated acceptor molecule as a substrate for iodinated or tritium labeled donor substrate. The number of assays for the DNA and RNA polymerases have been developed using the biotinylated DNA primers, DNA or RNA templates, and the tritiated nucleotide. Given their appeal as a therapeutic target, the various assay methodology have been approaches for high volume screening of viral polymerases and related proteins were recovered. For example, you can actually be able to use the SPA based transferase assay for the farnesyl transferase. So this is the assay what I am going to show, and what you are going to do is you are going to take the substrate, and then you are going to have the increase in signal, which means you are actually going to measure the product. In the signal decrease assay, you are going to measure the substrate. So substrate, as the substrate is getting cleaved, there will be a decrease in signal. Whereas in this case, there will be a transfer of the radioactivity onto the biotinylated substrate, and that's how you are actually going to have the increase in product. Then we have the product capture assays. So in the product capture assays, what you are going to do is you are going to do a time course analysis of the phosphodiester SPA based assay. So the time course analysis was performed using the human type 4 phosphodiesters incubated with 10 nanomolar cyclic AMP at 30 degrees Celsius. The reaction was stopped at time intervals by the addition of a 1 milligram of underivatized YSI SPA beads in 6 millimolar zinc sulfate. So you can actually be able to use the product capture assays, where you are actually going to have the substrate which is actually getting converted into the product, and then you can actually be able to add the antibodies, and you can actually be able to add the SPA beads, and this how you are actually going to see a signal. And when you add the SPA beads, it is actually going to terminate the signal, and that's how you are actually going to see a signal, right? And using this signal, you can be able to calculate the kinetics of the enzyme assays, you can be able to calculate the concentration of the products, and you can be able to calculate the other kinetic parameters as well. Then we have the assay design. So there are important factors what you are going to consider when you are going to design the SPA based radiometric assays. First is source of the enzyme, the second is design of the substrate. Then you are going to decide whether you want to use the signal increase or the decrease assay, which means whether you are going to signal increase means you are actually going to say whether it is easy to measure the product, or signal decrease means it is whether it is easy to measure the substrate. Then whether you want to make the on beat or the off beat, which means whether you want to capture the on signal or the off signal. Then you also going to have the optimization of the incubation concentrations. Then you are actually going to have the selection of the different types of SPA beads, and then you can also have the optimization of the amount of SPA beads what you are going to use for every reactions. And then you also going to have the performance criteria and the validation of the SPA assays. Then we have the solid phase versus solution phase SPA enzyme assays. So far what we were discussing about the solution phase SPA assays, where you are actually adding the SPA beads, and that's how you are actually going to collect the cap signal from the SPA beads, and you can convert that into the signal, and that's how you are going to convert that into the concentration of the substrate or the product. But we can also use the solid phase SPA enzyme assays. So the substrate is immobilized onto a surface. In this case, the SPA beads is in a solid phase enzyme assays. The enzyme is then free to work on the immobilized substrate. This format for assay design raises several points. First, the process can no longer be assumed to follow the Michaelis-Menten kinetics for a variety of reasons, the most important of which that enzyme and substrates are effectively compatibilized from one another. Okay, so there are many issues when you are going to do the solid phase SPA assays, because it is not giving the free movement of the substrate, and that's how they will not be able to follow the Michaelis-Menten kinetics. The substrate is concentrated at the bead surface, which is occupied a finite, constant, and especially a small volume in a assay tube. The enzyme, on the other hand, is assumed to be evenly distributed throughout the assay tube. As a result, the significant amount of enzyme will not be able to be used in the reactions, right? This means it is actually not going to give you the real picture of how you are going to use the the enzyme assays or the measuring the enzyme activity. It is also necessary to consider how the presence of the bead surface affects the kinetics. Interaction of the beads with the substrate and the enzyme must be characterized to study the kinetics of the enzyme. At non-saturating enzyme substrate concentration, the time course of the solid phase assay can be faster at a given quantity of substrate, as all the substrate is concentrated at the bead surface, and therefore the localized concentration is relatively high. So in this case, one of the major drawback of the solid phase is that the substrate is not evenly distributed. It's very concentrated. So the initial rate of reactions are going to be very fast because the substrate concentration is very high. But the later on, it is actually going to be very low because the substrate is, the concentration is going to be very low because it's already been consumed, right? So you are actually not going to get the equilibrium state actually. The solid state format provides a fast, simple and reliable assays for the drug screening, enzyme measurements or the purification using the less substrate. So one of the major advantage of the solid phase assay is that you are not going to lose the assay. You are not going to lose the substrate. So you can be able to reuse those beads again and again because they are immobilized onto the surface. So you can be able to even measure the activity. We can be able to reuse actually the same thing again and again. Then the solid phase assay, once validated, can be optimized to detect the inhibitor or more relative rates of the enzyme assays. However, not all enzymes perform well in solid phase SPA tests. This could be done due to this could be due to the active site's shape or the size of the substrate used. So this is very important because not all the enzymes are going to accept the substrate which is captured onto a particular solid support, because it is possible that the region what you have used for linking to the solid support may be very crucial for the enzyme to recognize the substrate, may not be for catalysis but for recognition of the substrate or binding of the substrate. And that's why some of the enzymes are relaxed, some of the enzymes are flexible, so they will be able to use, but not for all. Solid phase SPA assays. In a solid phase assay, all of the reaction components are present in solution, right? And the reaction can proceed in the absence of SPA bead. When the reaction has reached the desired level of substrate conversion, the assay is terminated in the presence of SPA bead. The product are then collected and the reaction rate can be calculated. This format eliminates the concern associated with the bead interference. Because the beads have a limited ability to bind the biotin, the assay must be designed with either an excess of bead or the known amount of bead to capture a constant proportion of the product. So this is a very important point, that you should have the enough amount of bead so that you should be able to capture all the substrate or the product, so that you should be able to give the real representation of the concentration of the substrate at that particular moment. If there will be a shortage of the beads, then the substrate actual substrate concentration is going to be very high, but the observation what you are going to get from the reactions are going to be on a lower side. So examples, there are so many enzymes what you can actually be able to use or what you can actually be able to use for enzyme assays. You can use the HIV integrase, HIV protease, HIV2 protease, CMV protease, endothelin converting enzyme, and so on. So these are some of the enzymes for which people have already developed the SPA-based enzyme assays. For example, you can have the kinase assay. So protein kinase are a type of enzyme that transfer the phosphate group to a specific protein, recognizes the sequence, thereby regulating their function. These enzymes are involved in a wide range of cellular processes, including cell growth, differentiation, inflammations, and are classified based on the functional property and cell location. These cellular mechanisms are crucial target for the therapeutic innovations. So for example, this is the target protein which is going to be recognized by the kinase, and it actually has a free OH group. So what will happen is, when you have the protein kinase, it is going to take this as a target, and it is actually going to have the radio labeled phosphate group, right? So this is the gamma phosphate, this is the beta phosphate, and this is the alpha phosphate, right? And this gamma phosphate is actually going to be broken down from here, and it is actually going to be transferred onto this OH, and that's why you are actually going to have the O phosphate, right? And that's why this is actually going to be the phosphorylated product, right? So phosphorylated protein. Now, if you have the scintillation beads, right, the scintillation beads are actually going to receive the beta particles, and that's how they were actually going to give you the lights. They are going to give you the lights, and that's how you can be able to use that. For example, is you can actually do the imaging of the MAP kinases, right? You can actually be able to use this, and you can be able to do the imaging of the MAP kinases. So the reaction mixture contained the 50 picomole biotinylated MBP. So MBP is a protein, the maltose binding protein. You can actually have the radio labeled ATP. So you see this is the radio labeled ATP, where you have the phosphate onto the gamma phosphate. Then you can have 25 picomole unlabeled ATP and 25 microgram ERK kinase. So this is the kinase what you are going to use in a MOPS buffer, right? And you can have the statosporin dissolved in DMSO was added to assay to convert the range, and assay was performed in 384 plates, and the reaction volume was 30 microliter. Our assay was incubated at 37 degrees Celsius for 30 minutes and all that. And then you are actually going to measure the activity. So in the experimental design, what you're going to do is you are actually going to do the amount of efforts put into the designing and optimizing the key parameters that influence the enzyme rates, and their measurement determine the robustness of any enzyme, right? Substrate concentrations and the purity, reaction volume, and in case of radiometric assays, the radiolabel selection and optimal specific activity. All of these factors must be considered carefully. So when you want to perform the radiometric assays, you have to design the experimenter very nicely so that you can be able to reduce the background of the reactions, and you can also be able to choose the different types of radiometric assays, radio labels, whether you want to use the tritium labeled or carbon 14 or any other molecules, right? Sulfur, right? And then you are going to use the technology. What technology you are going to use? Whether you are going to use the microplate technology or and ideally people are going to use the high-throughput screening assays, so we can actually be able to use the radio-labeled techniques for the microplate technologies. Then we can also see whether what kind of measurement techniques are available in your laboratory, so that also will going to decide what kind of radiometric assays you want to use. So you can actually be able to use the microbeta, which employs the dual photomultiplier PMTs and the classical coincidence counter and the top count. With a single PMT and a time-resolved counting approach can be used to determine the beta particle levels such as tritium level, sulfur, calcium, carbon, phosphorus, or the in a conventional scintillation counting, and as well as the 125I when used with the SPA. These instruments can be purchased with a variety of detector versions, allowing the sample in a complete 384 well plate to be counted in approximately 45 minutes, depending on the desired sample throughout. Then you can also be able to, because the radiometric assays just require the incubation setting of the reactions, and then you can actually be able to have some kind of separation technique so that you can be able to separate the radio-labeled substrate from the normal substrate or radio-labeled substrate from the product. And then you can be able to just have the technique to measure, right? So that's how you can actually be able to think about the automation of the assays. So the need to assay more sample will grow significantly in the pharmaceutical industries, particularly during the early stage of development. It is not surprising then that the much attention has been focused in the recent year on the improving the productivity of cost efficiency by the automatic or semi-automatic radiometric assays in the 96 and the 384 microplate formats. Then you can have the homogeneous radiometric assays such as the scintillation proximity assay can be automated using the wide range of commercially available robotic systems. So this is all about the radiometric assays. What we have discussed about what are the different ways in which you can be able to set up the radiometric assays and how you can be able to utilize or exploit the scintillation proximity assay based system to measure the activity of the enzyme without even going through the tedious process of separating the substrate or the radiolabeled product. So this is all about the radiometric assays. Let's now move on to the another approach where you are actually going to use the gel electrophoresis. So the gel electrophoresis is another approach where you are actually going to run the enzyme onto the electrophoresis, or you are actually going to use the electrophoresis to separate the labeled substrate from the non-labeled substrate or labeled product from the unlabeled product. So enzyme assays after the electrophoresis. So after electrophoresis, enzyme visualization consists of 5 stages. Stage 1, preparation of crude enzyme extracts. Then you require the electrophoresis. You can have the enzyme visualizations, documentation, and data interpretations. While the different electrophoretic method may produce similar separation pattern, the enzyme extraction from a biological source require the special considerations. So let's discuss the first step. First step is the preparation of the enzyme extract. See, the extract of the microorganisms, all of these techniques or all of these approaches we have already discussed when we were discussing about the extraction of the enzyme from the different hosts. So you can actually we have the rupture of the microorganism protocol. So you can use the different types of protease inhibitors so that there should be no degradation of the enzyme, and then you can use the extraction buffers, and then you can also use the procedures, and that's how you can actually be able to prepare the extract of the microorganisms. If it is an animal or the soft tissue, except in case of very small animals, specific organs should be used to enzyme extractions. If possible, use the fresh material. Otherwise, tissue blocks should be stored at 80 degrees. Before storing, the vascular system or organ should be cleaned with the 1.8% saline. Then you can also be able to have the processing of the mammalian bloods. So preparation of the mammalian blood serum protocol. So you can actually be able to follow the same protocol. This protocol we have already discussed when we were discussing about how you can be able to prepare the serum, right? When we were discussing about how to produce the antibodies. And the lastly we have plant tissues, right? So plant seeds, vegetables, buds, and cambiums are the most free of protein interfering substance and thus preferentially used for enzyme source. These tissues can be extracted using simple acid buffer or a neutral buffer, right? So 100 millimolar pH7. Such buffer can be used for herbs, right? So small plants, right? That are low in phenol, such as spinach or the PET. Then you can use the phosphate buffer frequently to retain the catalytic ability of the enzyme better than the tris, right? So as far as the buffer is concerned, you should always try to use the physiological buffer such as phosphate buffer rather than the chemical buffer like tris. And phosphate buffer on the other hand should be avoided when studying the metal ion dependent enzymes. And phosphate buffers are also should be avoided when you are trying to measure the activity of the kinases. Most plants store significant amount of phenolics in the vacuoles of their leaves and roots while having low protein content. So terpenoids and resin acids are also commonly stored. Unless protective agents are added to the extraction medium, when these compounds are released during homogenization, they denature the intrinsic protein. So that's why we should be very, very careful from the phenolics and tannins and all other kinds of secondary metabolites from the plants. So we should have a sufficient amount of the antioxidant molecules and the reducing agents so that they should not be able to affect the proteins. Now, once you prepare the extract, you can actually be able to run them onto the electrophoresis, and then you can actually be able to do the different types of visualization techniques. So, principle of enzyme visualizations. So, the basic idea behind the in situ protein isolation is to expose an enzyme to a solution containing the enzyme specific substrate. The enzyme is demonstrated when its catalytic action on the substrate results in a color reaction product. However, the primary reaction products are frequently colorless and require coupling with a visualization addend to produce a colored product. There are methods to visualize the oxidative enzymes. For example, dehydrogenases can be measured in two ways: by measuring the change in the fluorescence of pyridine nucleotide, NADPH or NADH involved in the reaction, or by visualization of the oxidation reaction using a tetrazolium site that turns colored when reduced. So you can actually be able to use either of these methods to measure or to monitor the dehydrogenases that are present in the gel. Either you can use the fluorescent method to measure to see how the NADH or NADPH is getting reduced, or you can be able to see whether you can actually run the enzymes, you can perform the reactions, and then you can be able to use the tetrazolium salt. For example, you can use the MTT, and that actually is going to give you the blue color wherever it is actually going to find the enzyme which is of the dehydrogenases. So oxidative reductive catalytic reaction according to the general equation is that it is going to take the substrate plus acceptor for hydrogen, and then it is actually going to give you the product plus acceptor which contains the hydrogen, and then in this process it is actually going to process the MTT, and that's how it is actually going to give you the color. A co-substrate O2 or an artificial indicator molecule can be used as an hydrogen acceptor. Oxidoreductases that transfer the hydrogen to oxygen are referred as oxidases, whereas those who reduce the pyridine nucleotide coenzymes are referred to as dehydrogenases. In the dehydrogenases to be visualized in vitro, the staining system must include a hydrogen transferring substance. Then we have the PMS that is the most commonly used substrate, and PMS has the ability to accept the hydrogen from the NAD plus and transfer it to a tetrazolium salt. Then we have the this kind of system where you have the substrate and hydrogen, and it is actually going to get converted into the substrate, and this hydrogen is going to be received or accepted by the reactive side, reaction side NAD plus, and that in turns is going to oxidize the PMS. And while the PMS is going to receive this hydrogen, it is actually going to give that hydrogen back to the tetrazolium salt, and that's how the tetrazolium salts are going to be get converted into the reduced form, and the reduced tetrazolium is actually going to give you the color. Then we can have the assay of oxidases. So the determination of oxidases and peroxidases is frequently being carried out using the 3-amino 9-entyl carboxyl as an electron donor, but the reagent should be used with caution because it has been shown in animal tested to be non-carcinogenic. Then you can use the 3,3,5,5 tetramethylbenzidine has been recommended as a substrate for so this is also short form, it is called as TMB, right? And the addition of the peroxidases and a chromogenic hydrogen donor such as 3-amino 9-methyl carboxyl can be used to visualize the enzyme reaction in which the H2O2 is liberated. So this is the carboxyl method where you can actually have the carboxyl, which is a yellow colored product, and it is getting converted by accepting the hydrogen after the reactions, and then when the peroxidases are converting the hydrogen peroxide into the water, they are actually taking up this molecule, and that's how it is converting a yellow colored carbazole into a brown colored carbazole, and these brown colored carbazole can be visualized. Then we have the methods to visualize the hydrolases, right? So hydrolases are the enzymes that catalyze the hydrolytic cleavage of CO, CN, CC, and all other kinds of bonds, including the phosphoric and nitric bonds. So the well-known method for detecting the hydrolytic enzymes are the use of substrate that produces a fluorescent compound when enzymatically hydrolyzed, and the use of naphthol derivative in substrate and coupling the enzyme hydrolyzed naphthol to a diazonium compound to produce a colored diazo dyes. The umbelliferone methods to detect the hydrolysis. This is the method what I have given to measure or to detect the activity of the hydrolases. And here what you are going to do is you are going to have the substrate. When it is being cleaved, it is actually going to be fluorescent, and that's how you can be able to measure the fluorescence. So then you can have the azo coupling method to detect the hydrolases. So as the substrate for azo coupling, derivative of alpha or beta naphthols are used. During enzymatic hydrolysis, the corresponding acid or amide or naphthol compound is liberated. The non-colored naphthol compound is non-enzymatically bound to a diazonium salt via the azo coupling process. The basic structure of diazonium salt is RNR, right, which confers color upon the formation to azo groups. The color of azo dye is determined by both the diazonium salt used and the compound to which it is coupled. Coupling of naphthol derivative to diazonium salt can be performed by the two methods. The diazonium salt is directly included in the test reaction, or it is added after the sufficient alpha or beta naphthol has formed. So these are the different types of assays which can be used by the the electrophoresis. And then we have the staining protocol. So once you have the staining protocol for the acid phosphatases, so you can have the equipments and the reactions, right? So you can have the starch gel, you can have the dye which is called fast blue, bronzanol blue, then you can have the citrate buffers, alpha-ethylene phosphate and acetones. So in this assay, what you're going to do is you're going to run the reactions and then you are going to do the staining protocol for the acid phosphatases. Then what you're going to do is you're going to dissolve the 10 milligram of phosphate garnet GBC salt in the 10 ml of citrate buffer pH 4.5 and add the 4 ml of orthonethylene phosphate, and you filter and drop on the cut surface of a processed starch gel. The appearance of blue band indicates the presence of an active enzyme. Note if the pH of the electrophoresis buffer is greater than the 7, incubate gel in the citrate buffer 30 minutes at 5 degrees Celsius before staining. Electrophoresis, you're going to run the starch gel with a pH 7 and 13 volt per centimeter 5 hours at 4 degrees Celsius. Then you can also use the measuring protein kinase assays. So remember that the protein kinase assay is actually going to do this, right? It is actually going to take the protein. Then you are actually going to add the ATP. So ATP is actually going to have the radio labeled, right? Or it is actually going to have the gamma phosphate, right? And then what will happen is that the protein is getting converted into phosphate, which is actually going to impart. So this is actually going to be either positively charged or neutral, whereas this is actually going to be negatively charged because it is actually going to have the phosphate group, right? So this negatively charged group is going to run towards the different side, whereas the positively charged group is going to run on the other side. So during a kinase assay, the protein kinase transfer a phosphate group from ATP to the substrate and impart a negative charge onto the substrate molecule. In the protein kinase assay, a peptide with a net +1 charge is incubated in the assay buffer containing enzyme, non-radioactive ATP. A controlled reaction is also performed where the enzyme is removed from the assay mixture. Both experimental and controlled reactions are loaded in the middle of the agarose gel. So the peptide present in the control reaction has the plus positive charge and it will migrate towards the negative electrode. Whereas the peptide in the experimental reaction has the net minus charge and it will migrate towards the positive electrode. The assay gives the qualitative or semi-quantitative information about the protein kinase assay. But this assay can be used for test different compound, to know where the optimal sequence of the substrate. The assay is easy to perform and it does not require any specialized equipment. So this is what you're going to do is you're going to take the peptide, which is actually going to have the net positive charge, and because it has a positive charge, it will run towards the negative electrodes. Whereas when it is getting phosphorylated with the help of the phosphate group from the ATP, the peptide is going to have the net negative charge. So it is actually going to have the -1 charge, and because it has a -1 charge, it will run in the different directions, right? So you're going to run the two reactions. One is you are going to run the control reaction. The second is you're going to run the experimental reaction. So in the control reaction, you are going to have the peptide plus ATP, right? So there will be no transfer of phosphate group from the ATP to the peptide, and that's why the peptide is going to remain as +1. Whereas in the experimental reaction, you are going to have the peptide plus ATP and with the enzyme, right? So you're going to have the enzyme. So because you have the enzyme, there will be a transfer of the phosphate group from the ATP, and that's why here you're going to have the -1 charge. And then what you're going to do is you're going to load these two in the middle of the agarose gel, right? So what will happen is that the control reaction, which is actually going to have the positively charged peptide molecule, will run towards the negative electrodes, whereas the peptide, which is from the experimental conditions, is going to have the phosphate group, right? So it is actually going to have the -1 charge, and because of that it actually going to run in this direction. And amount of peptide which is going to be migrate towards this side versus the amount of peptide which will migrate towards this side, because from this reaction it will not be 100%. So imagine that you have the 70% phosphorylations, 30% non-phosphorylations. So non-phosphorylated peptide will run in this direction, whereas the phosphorylated peptide will run in this direction. So if you measure the activity, or if you measure the intensity of these bands which are you're going to get on both the side, you can be able to calculate the activity of the particular protein kinase assays. So this is all about the enzyme assays, how you can be able to set up the different types of enzyme assays utilizing the different properties of the substrate or the product. So you can actually be able to use the photometric based assays where you are going to see how the substrates or the products are absorbing the lights. And then you can also be able to use the radiometric assays where you are going to use the radioactive molecules or the nuclei, and that's how you are going to measure the activity of the kinases, transferases and hydrolases. And lastly, we have also discussed about the electrophoretic method where you can actually be able to run the enzyme onto the electrophoretic gels, and then you can actually have the different types of staining methods to stain the substrate or the product, or sometimes you can also be able to have the flexibility of staining the cofactors, and that's how you can be able to identify the different types of substrates. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspects related to enzymes. Thank you.